Dear students, good morning to all. Hope you all are fine. Today's our offline class is about DNA fingerprinting. What is DNA fingerprinting? It is the technique used for determining nucleotide sequences of certain areas of DNA which are unique to each individual. DNA fingerprinting can distinguish one human being from another with the exception of monozygotic twins. 99.9% of base sequences among humans is the same. They have 0.1% of genome or 3 into 10 raised power 6 differences in the base sequence. The differences occur not only in genes but also in repetitive DNA. DNA fingerprinting involves identifying differences in some specific regions in DNA sequences called as repetitive DNA. Repetitive DNA. These repetitive DNA are separated from bulk genomic DNA as different peaks during density gradient centrifugation. The bulk DNA forms a major peak and the other small peaks are referred to as satellite DNA. Satellite DNA is classified into the following categories on the base composition: A is to T reach or G is to C reach, length of segment, and number of repetitive units. These categories are: number 1, VNTRs, that is variable number of tandem repeats or many satellites surrounded by conserved restriction sites. A small DNA sequence is arranged tandemly in many copy numbers. The copy numbers varies from chromosome to chromosome in an individual. The number of repeats show very high degree of polymorphism. As a result, the size of VNTR varies from 0.1 to 20 kb. Number 2, SSRs, single sequence repeats, or STRs, short tandem repeats, or microsatellites with 1 to 6 BP. These sequences normally do not code for any proteins, but they form a large portion of human genome. These sequences show high degree of polymorphism and form the basis of DNA fingerprinting. Polymorphism is the variation at genetic level since DNA from every tissue such as blood, hair follicle, skin, bone, saliva, sperm, etc. from an individual show the same degree of polymorphism. They become very useful identification tool in forensic applications. Further, as the polymorphisms are inheritable from parents to children, DNA fingerprinting is the basis of paternity testing in case of disputes. As polymorphism in DNA sequence is the basis of genetic mapping of human genome as well as of DNA fingerprinting, it is essential that we understand that what DNA polymorphism means in simple terms. Polymorphism, that is variation at genetic level, arises due to mutations. Allelic sequence variation has traditionally been described as a DNA polymorphism if more than one variant, that is allele, at a locus occurs in human population with frequency greater than 0.01. In simple terms, if an inheritable mutation is observed in a population at high frequency, it is referred to as DNA polymorphism. The probability of such variation to be observed in non coding DNA sequence would be higher as mutations in these sequences may not have any immediate effect in an individual reproductive ability. These mutations keep on accumulating generation after generation and form one of the bases of variability or polymorphism. There is a variety of different types of polymorphisms ranging from single nucleotide change to very large scale changes. For evolution and speciation, such polymorphism plays very important role. Thus, the basis of DNA fingerprinting is VNTR, a satellite DNA as probe that shows very high degree of polymorphism. The technique of DNA fingerprinting was developed by Alec Jeffreys. The technique, as used earlier, involved southern blot hybridization using radio labeled VNTR as probe. It included number one, isolation of DNA, number two, digestion of DNA by restriction into nucleases, number third, separation of DNA fragments by electrophoresis or RFLP, that is restriction fragment length polymorphism. Number 4, transferring or blotting of separated DNA fragments to synthesize to synthetic membranes such as nitrocellulose or nylon. Number 5, hybridization using labeled VNTR probe. Number 6, detection of hybridized DNA fragments by auto radiography. After hybridization with VNTR probe, the auto radiogram gives many bands of different sizes. These bands give a characteristic pattern for an individual DNA. The sensitivity of the technique has been increased by use of polymerase chain reaction PCR. Consequently, DNA from a single cell is enough to perform DNA fingerprinting analysis. This was all about today's offline class. Thanks.